Well, now we're going to talk about, we finished talking about the genetic of cancers from a very general perspective about oncogenes and tumor suppressors. Now we're going to talk about benign and malignant tumors. So key things that I want that you remember, again, this is one of the first classes. Later we will talk about this in much more detail. But the main idea is that imagine that you are growing this cell, yeah, and there is a lot of space. We are using cell culture, and this was a classic experiment that we will talk more in detail later. That it was you put a fibroblast because it's a very easy to culture, and as there is so much space in the right environment, what is going to happen is that the cell is going to make copies of themselves. So this is how we start studying how tissues regulate their cell numbers. And as there is space here, they will stop only until there is no more space. In other words, there is more space, so there is a signal here, and later we will study how these signals pass from one cell to another one. Then again, there is more space here. So what is going to happen? The membrane have receptors that allow them to go on and reproduce again. And finally, here again, the receptor said there is no more space. We should stop dividing. Okay, so this is what generally happens. Later in class, we will talk about this that is called contact inhibition. Yeah, so they inhibit the growth by contact, and we will talk about how there is new hypothesis, better models that talk also about mechanical tension that complement this model of how normal tissues regulate their cell numbers. Okay, and remember, if a cell died, yeah, so if one of them of these cells died here, what is going to happen is that it's going to uh, is going to be empty space. So what is going to happen? The space is going to be refilled. How? By a cell dividing again. Yeah, and in our words, let me show you. Let me delete this. So this cell died, as you can see here, is not there anymore. So here, the others, we cannot leave a space empty here. So cell division will occur. Of course, this is a very simple model. And for the people that have studied the comb rotation, we know that the cells are oscillating. They never leave a space. So in vivo, things are a little bit more complicated. And this is what we are studying and that we will cover later. So um, another thing that could happen, yeah, is not only that a cell died. Yeah, later we'll study the pathway of apoptosis and all this story. But here imagine that there is a mutation that can take place. So what happens when there is a mutation? That this mutation uh, can have multiple effects. Some of them will trigger cell death. Other ones, in most of the cases, don't have any effect. And if it's in an oncogene or a tumor suppressor, is when cancer can appear. Yeah, so mutations can occur spontaneously, yeah, and we already know some of the in the videos they describe why mutations can take place, yeah. So the environment can be a very important triggering of cancer. But what happened at the tissue level? Usually when DNA damage happen, the cells generally will die, yeah. So this cell is there, it will die, and now we are going to have a new cell. So in this way is how cells are repaired, and this is what generally happens in any tissue if there is a healthy tissue. Okay, so now that we know that mutations occur spontaneously and that cells generally repair them, so this is great. The only problem is that over time we will start accumulating more mutations. Yeah, so there will be mutations that they can just not be repaired because for example, if you have a mechanism like to repair the changes, but you have a mutation exactly in the machine that repair the mutations. Now, this will allow the accumulation of mutation, or you have a mutation in the mechanism that promote apoptosis. Now there will be not apoptosis. So in the long term, you will start accumulating mutations until one of those mutations become a into a cell that will produce a tumor, into a cancer cell. Yeah, and then this cancer cell will start proliferating. Yeah, and there are many mechanisms that can kill cancer cells. However, over time, you will start accumulating mutation, and this will outcompete the cancer cells will outcompete the normal cells. Yeah. So one way to increase the speed of this phenomena, to increase the rate of mutation, and it has been discussed in detail in the video, is through environmental factors like smoking cigarettes, some type of diets, UV radiation, and decreasing exercise. Yeah, so we already know that if we all this will just increase the probability of accumulating mutation faster. Yeah, so all of us, we're going to accumulate mutation. In these ways, we will accumulate mutation faster. And in the video, I'm sure that you listen to the guy talking about different type of mechanisms to do that. Okay, so now the next step. So we have this cancer cell that is there, but now it needs to proliferate and start out competing the other cells in the tissue and to form a tumor. But to have a tumor doesn't mean that you will develop cancer. Yeah, it means there are different type of tumors. Yeah, so this cell is going to, if you see, divide and start disorganizing the epithelium, continue dividing and divided until this become a tumor. Yeah, so a tumor, also known as a neoplasm, is an abnormal mass of tissue that may be solid or fluid filled. Yeah, so inside of this, it could be also just fluid, or it could be inside of that, it could have also cells. And later we will talk that they need a specific niche, tumors, and there will be a combination of cells. Um, so a tumor, as I said before, is not equal to cancer. There are two types of tumors that you have here multiple times. Ones that are benign and another ones that are malignant. The benign are tumors that remain in its current form, yeah, so like this. And the malignant ones are tumors that grow and spread. So these ones, the benign, is an early stage that if they accumulate more mutations, they can turn into malignant. Uh, so there is no way to predict, yeah, um, when a benign tumor is going to turn malignant. Yeah, we know that this process takes place over time, and depending on the tumors and the mutation that they accumulate, on the rate of mutations, many factors, this process could eventually happen. So the idea is, if you treat it here when they are benign, that is a generally a small surgery. I think most of us, if you have a mole and you go on to the doctor, they say, oh, this is a benign growth, so they will remove it. So not that is going to turn in cancer. Maybe will never turn into cancer, but it's a good way to prevent problems. Okay. So remember, benign, sorry, is the one that is the tumor that remains in its current form, so it's not going to spread to another places. However, the malignant is the opposite; it's going to grow and spread. So this is restricted in space, and this one is going to grow without restriction, without spatial restriction, and is going to move into a different regions. That this is when metastases take place. Okay, so two important type of tumors, benign and malignant. Okay, so now we're going to talk about two type of tumors. from this schematic, I want that you try to think about what are the difference, yeah? So look, one is enclosed in one region, the other one is not enclosed. Try to look for the nucleus, try to look for the shapes of the cells, yeah? Try to think about the mitosis, which one will have a higher degree of mitosis. So write it down, take some time. If you want, you can stop the video and then remember to upload this on Moodle. Okay, so I'm sure that you or stopped the video or wrote your ideas, but these are some examples between the benign and the malignant tumor. So first, there are well-defined borders, and here they have irregular borders. So here you see clearly that they are confined in a space. Here they are not confined in a space. And not only that, look, that we will talk about that later, they have these very regular shapes, yeah? In other words, this will be well differentiated, yeah, into one tissue. This will be poorly differentiated. Why? Because they look like different tissues, yeah? These irregular shapes, if you remember, there are um, these protrusions are like that look like fingers. They allow the cells to have motility. This is not normal. Yeah, it's like imagine the epithelium is like your house that is full. Each cell is like a brick. Imagine that there are bricks that start developing legs, yeah, feet, and start walking and escaping. So this is what is happening here. Cells are like in the epithelium in particular, they have some flexibility, but they are not going to move, yeah, like this. So this is will be poorly differentiated. So okay, remember, well-defined borders, well differentiated, and they you start getting more difference. Here they will have more irregular nuclei. Here you have irregular and large nuclei. If you remember, the HeLa cells were an example. When you start finding more differentiation, more difference between the cells, shapes, the nuclei, and if you remember the mitosis in that video, it was pretty interesting that here you will find less mitosis, while this one there is no control, so you start getting a lot of cells. Okay, so these are some. I talk about four characteristics between four difference between malignant and benign tumors. And there are more, but these four, if you remember those four, and of course this one are non-invasive, and this one will be invasive. These are the key features that differentiate benign and malignant tumors. Okay, so to finish this section, I told you in the beginning what is cancer, and I told you that it's not one disease, it's many diseases that are related. Yeah, why? Because they have uncontrolled cell division. Yeah, this is one trait, and the second trait is that they invade or spread or parts of the body. We will talk about many different features, but these are the two general that we talk about. And how it start? It start accumulating mutations that will misregulate the cell cycle, and it will end up producing a cancer cell. And there are two types of tumors. One that is the benign and the malignant tumor. And the difference, there are multiple morphological difference. And one of the keys that the benign will remain in his current form, while the malignant will grow and spread to other parts of the body. Okay, so now we talk, let me cross this one. So we already know what are tumor suppressors and oncogenes and how important they are. That not every single mutation is going to produce cancer, is mutation in specific regions. Then we talk about different type of tumors that are benign and malignant. Benigns remain in the space, malignant will tend to move. And now I'm going to give a review. Here I will, if you haven't took developmental biology or cell biology, I think it's a great idea that you watch this video, and I will add it later. So I will put it in a different link. If not, if you already took all those courses and everything is fresh, go and take the quiz. And if you have any doubt, you can watch the video.